Good afternoon, guys. So today I'll be talking about my project, which is the development of the PDX humanized mouse model for high-grade serous ovarian cancers. So some basic infos about ovarian cancer. So from a Canadian Cancer Society in 2023, so it is estimated around 3,000 of Canadian women will be diagnosed with high-grade serous ovarian cancers. With eventually about 2,000 of them will died from it. So it's mainly originated in the fallopian tube instead of the ovary. So they accounted for around 70% of all the ovarian cancer cases with the worst prognosis. Almost all cases, they have TP53G mutations, and 20% of all the cases, they have BRCA1 and BRCA2 mutations. These eventually lead to the deficiency of the homologous double-strand DNA repair deficiencies in the patients. So why we chose to do humanized mice is because the advantage of humanized mice model gives us the abilities to mimic the human tumor microenvironment. It also allows us to create a natural tumor heterogeneity when we look at the tumors. And it also acts as a preclinical model for the drug responses if we want to test new drug therapies on high-grade serous ovarian cancers, especially BRCA1 and BRCA2 types. So developing such a model allows us to examine the interactions of the human immune cells, especially the T cells or B cells with the high-grade serous ovarian cancers. And we can also investigate how they progress in a live model and test for therapeutics responses. So the aims of the project is that we try to establish the human CD45 cells in the mice by transplanting hematopoietic CD34 cord blood cells from human. And then we also try to investigate the optimal setup, which is how many cells we should inject to the mice to create optimal humanization models. We also try to establish the human high-grade serous ovarian cancers PDX in such humanized mice. And for the last point, we try to compare and categorize the histology of the high-grade serous tumor that grows in such humanized mice and the non-humanized mice model. So the first thing first, which mice strain we chose to use. So we chose to use a mice called ENW mice. So they are genetically identical to NRG mice, except that they possess of the human interleukin-3, the human granulocyte macrophage stimulating factors, and the human stem cell factors. And they also have absence of a normal W41 gene. So such setup allows us to support the multi-lineages of the engraftment of the human cord blood cells into the mice. And also compare with other mice, we don't have to do the radiations to get rid of, to provide the niche for the engraftment. So without doing irradiations on the mice could have a better lifespan and better health conditions for the coming up experiments. So the setup is we get, so this is an early stage setup. So we have 7 ENW mice at the beginning, and then we injected human cord blood cells to them in 5 of them, and then 2 is non-injected cells. So we proceed with bone marrow aspiration at week 6. And then we collect saphenous vein blood from week 7 onward towards the final stage of the experiment. With all the samples that we collected, we proceeded to reprocess it and proceeded to the flow cytometry analysis. So for the panel here, we only use a quick CD45 panel for now, because we only want to check the setup and want to check the humanization levels in each mice. So such things repeated every week until the experimental endpoint, which is week 14 of the study. So this is the flow panel and the gating strategy that we used for the bone marrow aspiration we did on week 6. The flow panel we used included human CD45 antibodies. We used 2 different human CD45 antibodies because they target different epitopes of the human CD45 cells. So by looking at the 2 antibodies together, we get a more comprehensive readings of the human CD45 percentage cells in the mouse bone marrow. So so this is the gating strategy we use for the bone marrow. The result as follows. So we injected 5 ENW mice with CD34 hematopoietic cord blood cells, and 4 of them show a very strong and robust engraftment levels, which is represented by the orange bar, which is the double positive CD45 human CD45 cells. They have at least 30% of populations in the mouse bone marrow. which is quite strong and robust. To consider as a robust engraftment, we need at least around 20% of the percentage in the bone marrow. So we certainly did it. So for the saphenous vein blood that we started to collect from week 7 towards week 14, here's the panel that we did. Generally, it's the same, it's the similar as that we did in bone marrow, but we added one more different antibody, which is the mouse CD45 antibodies to the panel. So the reason that we're adding mouse CD45 antibodies is if we want to get pictures of the human CD45 versus the mouse CD45. So by looking at two different numbers, we can actually calculate the humanization levels of each mice. So so that's why we included one more antibodies in the saphenous vein blood panel. So I won't go through every week's data. So I picked 3 weeks from it. So which is week 9, week 11, and week 14. So out of the 5 ENW mice that we injected with the CD34 cord blood cells, 4 mice were successfully humanized and maintained a very stable, robust humanization throughout the entire study, which is until week 14. The humanization ranged from 15% to 40%. Also note that when we look at blood samples compared to bone marrow samples, the humanization level is always lower in blood compared to bone marrow, around 20%-ish. So to conclude that we still get a very robust and humanization maintained throughout the entire study. So why did we choose to put week 14 as the last week of sampling of the blood? It's because that multiple mice start to develop different sickness starting from week 14, and their condition is not available for us to take more blood. So from week 14 onwards, we tried to rescue them, but we couldn't. So we decided to make week 14 as the final stage of the sample collections. So, yep. So from week 14 onwards, we observed graft versus host disease in the mice. So they start to lose weight, they start to show some behavioral changes. So these are the 4 mice that develop different GVHDs. So we can see a significant weight loss starting from week 14 onwards. So besides looking at weight, we also look at histology slide. So we look at the H&E slide of how the GVHD actually occurs in these mice. So we look at lung and livers, and then we can see a lot of things going on in the GVHD mice, such as we see extended areas of necrosis in the liver. We also see wall thicken in the lung as well. Besides looking at H&E slide, we also look at IHC slide. So we stained human CD3 cells and human CD45 cells in both lung and liver. So yeah, we can see a lot of positive signals in both the humanized mice compared with non-humanized mice. So yeah. So the future work will be we will inject the tumors in the next batch of humanized mice, which we already did, which is now waiting for the sampling period. So we will also characterize the tumors when we get the tumors out from the mice. So yeah. So the conclusion is that we successfully generated 4 humanized ENW mice for now, and then we also figured out the cells, the numbers of cells we should inject to each mice, and then the time point, etc. So we have also investigated the GVHD situation in all mice. So yeah, so that's all for the presentation. Thank you so much.
Okay. Hi, everyone. My name is Natalie. I'm one of the PGY5s in the OBGYN program here at UBC, and then I'm going to be starting my Gynae-Onc fellowship next year. So I'm going to be presenting on chemotherapy response score as a predictor of complete cytoreduction at interval debulking surgery in high-grade serous ovarian cancer. My supervisor is Dr. Brar. I have no conflicts of interest. So that was a nice segue that we just had. As many of you are aware, epithelial ovarian cancer has the highest mortality rate of all the gynecological malignancies, and high-grade serous carcinoma is the most common histological subtype. So the cornerstone of treatment for high-grade serous remains surgery, either preceded or followed by platinum-based chemotherapy. So you can either have what's called a primary debulking surgery, where when the patient presents to care, they have a surgery trying to remove all of the cancer at the very beginning of their treatment, and then they will have either 6 to 7 cycles of chemotherapy following this. Other patients who are it's deemed that their surgery is high morbidity, possible mortality, will need a bowel resection, that sort of thing, will have what's called neoadjuvant chemotherapy prior to their surgery, and then they'll have what's called an interval debulking surgery, and then have more chemotherapy after their surgery. It's somewhat controversial, but they have done randomized control trials to say that neoadjuvant chemotherapy followed by interval debulking surgery is shown to be non-inferior in overall survival in these randomized control trials. So we also know that optimal debulking at the time of surgery, whether it's the primary debulking surgery or the interval debulking surgery, those patients have the best outcomes and the best overall survival, meaning that they live the longest. We also know that patient selection for these interval debulking surgeries is crucial. So those who aren't optimally debulked have poorer outcomes, but they also have the added morbidity and mortality of a laparotomy, so an up and down incision on the abdomen, time in hospital, time away from their family, and time away from chemotherapy, possibly like another kind of chemotherapy that may provide better treatment. There are several studies in the literature talking about ways to predict which patients will be optimally debulked at the time of surgery, including laparoscopic scoring systems, radiological scoring systems, patient factors, that sort of thing. In terms of background that we need to know for this study, in terms of, chemotherapy response score is a pathological scoring system that evaluates tumor response to chemotherapy and has been emerging as a potential prognostic tool in high-grade serous cancer. So in brief, a CRS score of 1 is showing that the tumor has no or minimal tumor response to the chemotherapy, whereas a CRS score of 3 shows a complete or near complete response on pathology scoring system. So in this study, we sought to determine whether CRS from tumor samples after neoadjuvant chemotherapy, so that primary chemotherapy that the patient had, and prior to their interval debulking surgery, could be a useful prognostic tool in selecting which patients would be good candidates for surgery. So for example, a CRS score of 1 after someone had 3 cycles of neoadjuvant chemotherapy might indicate that this patient would be unlikely to benefit from an interval debulking surgery. Instead, they could have more cycles of chemotherapy, alternative plans could be made, and maybe save this patient quite a bit of morbidity and possible mortality. So we performed a retrospective chart review of 787 patients with ovarian cancer from 2010 to 2020. In the end, we identified 87 patients who met the criteria to be included in the analysis. This is primarily limited by patients that had a CRS score assigned to them by the pathologist. We then performed a multivariable logistic regression on the identified group, adjusting for CA-125, which is a tumor marker, a blood test that we do prior to surgery. Again, we were limited by the smaller number of patients that could be included in this final analysis, and we couldn't include other variables. So just looking at this, on average, each unit increase in CA-125 prior to neoadjuvant chemotherapy was associated with a 5.6 times the odds of an incomplete debulking, making it a good confounding variable here. Our results show that compared to a CRS score of 3, patients who had a CRS score of 1, so I would call that a worse score, had 15.8 times the odds of having a partial or suboptimal debulking, meaning they could not take all the cancer out after controlling for CA-125. When compared to a CRS score of 3, patients who had a CRS score of 2 had about 8.7 times the odds of an incomplete debulking after adjusting for CA-125. This is just a written description of that. So our results indicate that laparoscopic tissue sampling prior to interval debulking surgery may be helpful in selecting which patients will benefit from surgery and may prevent unnecessary open-close laparotomies, high morbidity, and possible mortality associated with surgery. This could be combined with laparoscopic scoring systems and would require multidisciplinary teamwork to allow for tissue samplings either by IR or for biopsies and potentially laparoscopy. Of course, further studies with larger samples are needed to further evaluate this. Questions? Thank you.
Hi everyone, I'm Juliana. I'm a PhD candidate at David Huntsman Lab, and today I'm being presenting my research. Copy number signatures identify therapeutic opportunities for p53 abnormal endometrial cancers. So endometrial cancer is the most common gynecological cancer in North America, and I'm studying the p53 abnormal subtype, which is a non-pooling mutant, mismatch repair proficient, but has an abnormal p53 IHC staining or a pathogenic p53 mutation. And although it only counts for roughly 15% of our endometrial cancers, it is the most aggressive subtype, accounting for 50 to 70% of the endometrial cancer mortality. Therefore, therapeutic advantages are needed. So our aims were finding biomarkers to better stratify the subtype, and with that, to improve the treatment with targeted therapies. So for that aim, we used a couple methodologies. One was shallow whole genome sequencing, which is just genome sequencing, but roughly 5 times the coverage for us. Target panel sequencing with 275 cancer-related genes, and we derived copy number signatures from the shallow whole genome sequencing data. So this is an example of one of our samples with the copy number profile from the shallow whole genome sequencing. In the x-axis, you can see all the chromosome numbers, and the y-axis, the log change in the chromosome number. So at 0, it's a normal copy, and if it's more or less, it's like doubled or lost copy number, and you can see those cancers, they have high loads of copy number alterations, and in this particular sample, we have a couple amplifications, especially HER2 and Signe. So we have roughly 200 samples, a little bit less than that, in our cohort, and we profiled copy number for all of them. And with that, we derived signatures using as base this paper that was published by the Brenton Group that did copy number signatures using shallow whole genome sequencing, but in ovarian carcinomas, high-grade serous ovarian carcinomas, which are also p53 mutant. So using a similar pipeline diagram, we identified 5 copy number signatures in our cohort. One was associated with BRCA1 and 2 loss, and diploid tumors, which we call our HRD, homologous recombination deficiency signature. Our signatures 3 and 4 were both correlated to a haploid state, cyclin amplification, HER2 amplifications, and MYC amplifications. The only difference in between them and signature 3 was also correlated with PIK3CA mutations. And for our signatures 1 and 2, we couldn't find any molecular associations. The only thing we found was the endometrioid histotype was association with signature 2. Our targeted panel results, so beyond p53, of course, the top 5 mutated genes were PIK3CA, PIK3RNA, PIK3R1, FBXW7, and PTEN. But as you can see, those tumors don't have a high load of mutation burden. It's more like a copy number related cancer. So we put all those data together, our copy number signatures, the amplified oncogenes we found, and our mutations. And we were able to find a targeted therapy, a possibility of a targeted therapy in 75% of our cohort. So either have one or multiple markers. And the markers we found were HER2, amplification on overexpression, HRD, homologous recombination deficiency, either for the tumors that were in our signature 5 or had BRCA1 or 2 mutation, cyclin overexpression or amplification, and also FBXW7 and or PIK3RNA mutations were also found as a possible target to use in those tumors identified with those alterations. And we are now following up with those markers in our lab, both using cell lines and PDX model systems to assess how useful that would be in our context of p53 abnormal endometrial cancers. I don't have much time to discuss all our data, but this is now a published research. So if anyone wants to look more on that, it's now published at Clinical Cancer Research. And I would like to, my acknowledgements go to the Huntsman Lab and all people there, especially David, Dawn, Jessica, McAlpine, and all the colleagues that helped me with that. Thank you. I'll take questions.
Hello, my name is Ran, and I'm an undergrad co-op student under the supervision of Dr. Yin Wei. I will be presenting some results I obtained supporting MCT1 inhibition synergizes glutamine metabolic inhibition to suppress the growth of SMARCA4/2 deficient ovarian cancers. For background, the focus today will be on small cell carcinoma of the ovary hypercalcemic type, or SCOOT in short, which is a highly aggressive and a relatively rare cancer type. On the histochemistry images here, you can see it is characterized by a diffuse sheet-like architecture of small cells, and as pointed by the arrow, you can see follicular architecture as well. At the genetic level, they are characterized by concurrent loss of SMARCA4 and SMARCA2, which are mutually exclusive ATPase subunits of the Swiss-Nif chromatin remodeling complex. This is relatively uncommon and thus leads to a distinct phenotypic profile in SCOOT. So from previous research, we have observed that SMARCA4/2 deficiency caused decreased CLOT1 level in both SCOOT patient tumor samples and cell lines. And as shown from the Western blot and immunohistochemistry images here. So CLOT1 is the main glucose transporter on the cell membrane. And what this decrease in CLOT1 translates into is that you can see decreased glycolysis and increased oxidative phosphorylation, as shown by the Seahorse assays. Looking closer to this reason behind increasing OXPHOS, we observed significant increase in glutamine transporter SLC3882, suggesting these cells might have used glutamine as the alternative main source of energy to drive the TCA cycle and thus leading to increasing OXPHOS. To exploit these characteristics, we tested whether blocking glutamine metabolism would inhibit SCOOT cell growth. So we chose two different therapeutic options here, alanine, which is a competitive substrate for SLC3882, and CBA39, which is the GLS1 glutaminase inhibitor. And when we tested them in vitro and in vivo, we found both are able to reduce SCOOT cell growth. However, the results on the right here, you can see the patient-derived xenograft mice model is a bit inconsistent in that in the right top, it is extremely effective in inhibiting, but on the bottom right, maybe not as much. So we are developing ways to improve this independent individual treatment here. When we consider the TCA cycle again, you can see glutamine is not the only way to feed into the TCA cycle. For example, pyruvate is an alternative nutrient that can go into it. So there are two main ways the pyruvate can be produced in the cell. The first way is through glycolysis, which is already significantly reduced in SCOOT. So this leaves us with the second way only, which is through direct import from the external environment through monocarboxylate transporters, or MCTs. So this is a family of proton-link plasma membrane transporter that directly import lactate, pyruvate, and other carbon source. So we hypothesized that in blocking MCTs, in addition to glutamine, we would limit the growth of SCOOT. So here we selectively chose MCT1, which is the main pyruvate carrier, as our target. And AZD3965 is the drug that we will be using, as it has shown clinical efficacy in previous research. So this leads to our hypothesis that MCT1 inhibitor AZD3965 will inhibit cell growth synergistically with alanine, a potent glutamine uptake inhibitor. And we also would do with CBA39 as well. So here we first validated our assumption that exogenous pyruvate indeed act as an alternative nutrient for SCOOT. So in this setup, we added alanine and CBA39, limiting glutamine metabolism. In addition to that, we added sodium pyruvate into the cell culturing media. So as shown on the graph, adding 0.5 millimolar of sodium pyruvate is sufficient to increase the endpoint confluency, showing presence of extracellular pyruvate can indeed impede with alanine and CBA39. So this results support that blocking pyruvate transporter MCT1 can be a feasible option in co-treating with glutamine metabolic inhibitors inhibiting SCOOT growth. So now we moved on to conduct panels of different drug combinations, here with alanine and AZD3965. So the synergy plots shown here are generated after 3 days of treatment with alanine and AZD3965 with various concentrations. And they are based on the endpoint confluency. So to interpret the graphs, you can see on the x-axis and y-axis, you see alanine and AZD3965 concentrations, while the z-axis represent the HSA synergy score. So for reference, a good HSA synergy score is above 10, and pretty much the redder the color, the better it is. So we see here that in SMARCA4/2 deficient ovarian cancer cell lines, they have large red areas, suggesting this is a possible treatment. And on the right here, for the control for the SMARCA4/2 proficient cell line, you can see they are marked by green, which suggests the drug is antagonistic in this case and suggests tumor specificity. So similar result trend is seen for CBA39 and AZD3965, where large synergistic areas are observed when AZD3965 concentration is above 15 nanomolar. So to directly observe the efficacy of the drug combination, I graphed selected concentrations of alanine and AZD3965 concentrations here. So you can see at 6.25 grams per liter alanine and 80 nanomolar AZD3965, both are able to decrease the endpoint confluency of SCOOT by around 50%. This is not a bad number, but however, as you see, the drug combination, both at lower dosage, are able to achieve a stronger effect together. So this is great news that we can minimize the amount of drug needed for future in vivo and clinical trials. For conclusion, pyruvate supplementation compromises the effect of targeting glutamine uptake and metabolism in SCOOT cells. This is the main reason we moved forward with blocking MCT1 and selecting AZD3965 to couple with alanine and CBA39. And our drug combination shows high synergistic value above 10. Our future experimental directions would involve targeting these treatment in SCOOT cancer cell lines in targeting in organoids and animal models of SCOOT and look into their effects in other SMARCA4/2 deficient tumors as well. Thank you so much for listening, and thank you to the ones listed here as well as other people in Yemen lab and Huntsman lab. Thank you.
Endometrioid ovarian carcinoma, or ENOC for the short, is the least studied form of epithelial ovarian cancer. It's a completely different disease than the other types or histotypes, but still there is a lack of specific guidance for ENOC. Different from the most common one, the high-grade serous, ENOC usually affects younger women that may have more concerns about fertility issues. So in this context, over-treatment, ineffective treatments are still a big concern. My name is Bianca, and endometrioid ovarian carcinomas are the main focus of my postdoctoral research at Dr. Inglesi's lab, and I'm going to talk about how we can improve prognostication for endometrioid ovarian carcinomas using hormone receptor expression. So ENOC can be further divided in 4 different molecular subtypes. I think you guys already heard today. It's the same classificatory system developed for endometrial carcinomas in the uterus, but we are talking about ovaries right now. So the molecular subtypes, each one has a specific molecular alteration that can define them. They have a clear separation of prognosis, except for the NSMP. So all the cases that didn't fit in the previous subgroups were put all together in this no specific molecular profile subtype. That's why the subgroup is so heterogeneous, not only regarding molecular structure, but also regarding prognosis. And this is the subgroup we are interested in the most, because we believe if we can further divide the subtype, we can help to provide clear information that can help and assist better decision-making for these patients. We have an established cohort of more than 700 patients from 5 different countries, and all the ovarian samples were assembled in tissue microarrays, the DNA was extracted, and we did immunohistochemistry analysis, mutation analysis, especially including the biomarkers and the genes that are required for this type of molecular classification. And finally, we evaluated the prognostic value of estrogen receptor, progesterone receptor, and beta-actina on survival outcomes. First, what we saw, as expected, the majority of the patients were classified as no specific molecular profile subtype. As you guys can see over here, the subtype has an intermediate prognosis, but we know this is diluted because due to the molecular heterogeneity of this group. So this classificatory system, it is still not enough for ENOC, because we are talking about more than 70% of the patients, the majority of the cases here falls into a subtype that we know the information for it is still elusive. When we look at the biomarkers, we can see that the patients who are estrogen receptor positive, they have a better prognosis than the patients who are ER negative, and this happened in the full ENOC cohort and in the NSMP subtype. For the progesterone receptor, we saw similar results, so the patients who are PR positive, they do better. And for beta-actina, we also detect a prognostic association, but the size of the effect was smaller. In the multivariable analysis, adjust for age, stage, and grade, we could confirm the prognostic value of these biomarkers, especially in the full ENOC cohort, and in an NSMP subgroup, only the progression-free survival remained significant for the 3. But what happens if we look at these 3 biomarkers at the same time? They happen in combinations. So we almost never see a patient that is only PR positive or only beta-actina positive. In the full ENOC cohort, the majority of the patients were dual positive for ER/PR, while in the NSMP subgroup, the triple positivity was the most common combination. So if we compare between the most frequent combinations, what we see is the patients who are ER/PR positive, regardless of having beta-actina positive in purple here, or beta-actina negative in blue, they are quite similar regarding survival, but they have a better prognosis when compared with the patients who are ER positive only or triple negative. And this happened in the full ENOC cohort and in the NSMP subgroup as well. Again, the multivariable analysis confirmed this. When we compare the triple positive with the dual positive, there was no significant difference regarding survival. But if we compare these 2 groups with the patients who are ER positive only or are triple negative, the presence of dual positivity ER/PR was correlated with better survival outcomes. Based on that, we propose a further division, especially for the NSMP subtype, between the patients who are hormone receptor positive. They are probably good candidates for de-escalation of chemotherapy, for example, because we know they have a better expected outcome. We can probably avoid to overtreat them. While the patients who are hormone receptor negative, we definitely need more research to find the best option of treatment for them. I'd like to express my gratitude, especially to the patients that by accepting donated tissue, they make this type of research possible to happen, and everyone that make this research possible to happen and continue to happen. Thank you very much, you guys, for sticking around and for the attention. Thank you.
Hello, everybody. This is Mariam. Today, my presentation is about domain generalization in deep learning for multi-center histopathology cancer diagnosis. I will present today with a focus on ovarian cancer. Ovarian cancer is the deadliest cancer of female reproductive system. Every year, thousands of women get diagnosed with this type of cancer, and unfortunately, many of them cannot survive. There are 5 common histotypes for this type of cancer, each with distinct molecular information, different clinical outcomes, and therefore different treatment. So accurately classification subtypes of ovarian cancer is very crucial for the effective treatment. We need expert pathologists in gynecology for diagnosing ovarian cancer. However, general pathologists are more available, and their inter-observer agreement for diagnosing ovarian cancer is quite moderate. It's about 0.60 Cohen-Kappa coefficient, while this number is about 0.89 for general pathologists. The challenge is that most of the underserved community have lack of access to gynecopathologists, and even well-developed communities face shortage of gynecopathologists. So this situation highlights the need for an AI model to learn from gynecopathology that can be used globally across the world. Integrating AI with clinical pathology has enhanced the diagnostic accuracy and efficiently, ultimately improving the patient outcome. Our goal in this study is to develop AI models to learn from data from one center and then deploy these models globally across other medical centers. However, here we are facing a big challenge called domain shift, which means that samples collected from different centers are having variations in their color space. This is not a big deal for pathologists to deal with this inconsistency, but AI models tend to provide good results for samples from one medical centers, but struggle to provide satisfactory results for other medical centers. To address this issue, we proposed ADA, stands for Adversarial Fourier-Based Domain Adaptation. In our model, we have two main parts of Fourier-based enhancer and adversarial part to encourage the model to learn features and adapt the features to be less sensitive to the source of data. For the sake of the presentation time, I will not go through the technical details of the model, but would be very happy to answer any question regarding that at the end. We tested ADA on 3 sets collected from 3 different centers. Our discovery cohort includes samples from VGH, and we use samples collected in Calgary and Netherlands as 2 different validation sets. In the discovery cohort, we could achieve the balance accuracy of 80%, and for the validation set, in average, we could get 76% of balance accuracy. And for all data sets, we could surpass the performance of other state-of-the-art AI-based methods during the last couple of years. We also showed that our ADA model could provide satisfactory results for majority of the subtypes of ovarian cancer, except the Endometrioid carcinoma, which were more frequently misclassified as high-grade serous, and this is a known challenge for pathologists as well. So let me tell a story that how AI and pathologists can benefit from the knowledge from the other side. We shared the misclassified slide of the Netherlands cohort with the pathologists to review them for the second time. As the result, 4 cases were excluded, and the labels for 16 cases got revised. Surprisingly, 11 out of those 16 cases got revised to the output of the AI model. And as additional experiment, we asked the pathologist to provide more slides for the misclassified patient, and using the new slides, we could improve the performance to 85% of balance accuracy. Let me evaluate the performance of ADA in several visual ways. In the left part of the slide, you see the slide level heat map. The first column shows the ground truth label, tumor annotation done by pathologists. The other column shows the AI-based, like different AI-based methods prediction on this slide. The last column is corresponding to the ADA performance. The highlighted area is the most suspicious part, the most relevant part of the slides related to that specific type of cancer. And we show that the output of ADA match perfectly with the tumor annotation done by pathologists. In the right part of the slide, we show the top patches, the most relevant patches selected by ADA method to the specific subtypes of the slide, and confirmed by pathologists. All of these patches have tumor-specific information related to that subtypes. And in the last figure, we compare different methods and show that ADA was successful in separating different subtypes in the feature space. It's worth mentioning that providing this visual information could help the pathologists to improve the performance. It has been proved in several research. We expanded this area of research and organized ovarian cancer subtype classification and outlier detection competition, the Ocean Challenge, hosted by Kaggle. Ocean became a really good landmark in histopathology research by attracting the largest number of participants among all competitions in this field for the last couple of decades. And also with the Ocean, we introduced the largest and the most diverse data set of ovarian cancer histopathology image. It is also worth mentioning that AI was really successful on the other type of cancer, including bladder, breast, and pleural cancer. ADA has been accepted in Precision Oncology Journal. At the end, I would like to thank OFCARE team, pathology team, both at UBC and University of Amsterdam, BC Cancer, GCI group, and special thanks to AIM Lab members, especially my supervisor, Dr. Ali Bashashati. Thank you very much for your attention, and I'm happy to answer your question.